everyone, welcome back. So for this week, we'll be doing something a little bit differently. So our lab is still kind of shut down due to the COVID situation. So I will not be able to perform the BCA assay right now. But to kind of keep, keep with the schedule, I'm still going to try to lecture about it. And for this week, instead of having a lab report, all you guys will have to do is do the BCA quiz. So later on, I will still record the actual experiment. This is so that you guys will have it for yourselves when you make your final presentations. But for now, just remember that all we'll be doing, or all you will have to do this week is just do the BCA quiz. So remember 2 weeks ago, we finished up our purification. We purified our beta-gal off of an anion exchange column, and then off of the size exclusion column. And when we looked at the gel, we saw that our beta-gal was very, very clean. So this is a very good indication that we got as much beta-gal as possible. We saw that from the very thick bands. And that if we have any contaminants, it's probably very little to nothing. So this is good. So then the next thing I would do is I would take all of the fractions that had our beta-gal sample and concentrate it down to 500 microliters. So in these 500 microliters, I need to know how much protein is actually in there. What kind of concentration are we working with? So that's what we're going to try to do this week. We're going to try to figure out what or how much protein is in this solution. And there's a couple different ways we can do it. The first one is the very simplest way. It's using just the UV vis on our sample. However, that can give us unreliable results. We can also try to use fluorescent. We can use a fluorescent dye that will bind to the protein, and the more dye that binds, the more brighter the actual solution will appear. So this is a very good way to kind of check the protein concentration. But it's expensive and we don't really have the machinery to do that. So that leaves us with two other options, a colorimetric assay or a Biuret assay. So both these assays kind of utilize the same mechanism. It really just kind of measures color changes depending on the amount of protein in the solution. But one does it in a direct method and the other does it more of an indirect method. So the BCA assay, which is a Biuret assay, will actually be doing the protein analysis using an indirect method. And I'll talk a little bit more about that as we progress. But here you can kind of see what BCA looks like. It's a big, clunky molecule. And right here, these two nitrogen groups are what's going to be important about BCA. So like I said, BCA is an indirect method to determine protein concentration. It basically allows us to see differences in concentration by the changes of color, and we can actually analyze these changes of color on a UV vis at 562 nanometers. So here you can see about 8 tubes set up, each one having a different concentration of a protein, let's say 1 mg up to 8 mg per ml. And you can see that as we go up higher in concentration, the color of the solution gets darker purple. When we place this on a UV vis, we're able to actually measure the absorbance, and we can compare it to our sample and figure out where our sample kind of lies on this graph. So the actual way that this assay works is it's based on the reduction of copper 2+ to copper 1+. So remember, I said a colorimetric assay would be the direct method to doing it. For that assay, we would really add a dye that binds specifically to the protein. But for us, that's not really applicable. We can't really use the dye because it could give us bad results. So we found that the BCA assay is a little bit more sophisticated and it's able to give us very, very precise results, which is why we want to do this one. So the way that we get this reduction from copper 2+ to copper 1+ is by adding our protein. So what ends up happening is the protein in the sample, if there's any copper 2 plus present, it will reduce the copper 2 plus down to copper 1 plus. And so you can kind of see that the more sample, or the more protein that is in our sample, the more we're going to form copper 1 plus. Once copper 1 plus is present in the solution, our BCA molecules are able to chelate that copper 1 plus molecule. So here you can see that we need 2 BCA molecules for every 1 copper 1 plus. And you can see that the chelation happens right here at the nitrogens. Once this formation occurs, once we get this copper BCA complex, that is what's going to cause the solution to turn purple. So this reaction, this formation of the complex, it really just can occur even at room temperature over a long period of time. But what we like to do to kind of speed up the process is we actually throw it in a heat block at 60 degrees Celsius to just get this reaction to happen faster so that it occurs in maybe 10-15 minutes. So here I kind of wanted to give you a little diagram of what's going on. So in the top, we have about 8 tubes that each have a different known concentration of a protein that we, I guess, absolutely know. So for us, that's BSA. We use BSA because it's rock solid. It does not ever break. It's easy to store. We have a ton of it. So it's easy to measure. So we can very easily have a very accurate standard curve based on our BSA. So for each of these tubes, we add a different amount of BSA. So 1 mg per ml, 2, 3, 4, all the way up to 8 mg per ml. And we also prep a tube that has our sample in it. So remember, we don't actually know what the concentration is. Once we finish making up our tubes, we then throw them in a heat block at 60 degrees and just wait 10 to 15 minutes, and eventually the tubes will turn purple. So remember, the more purple the solution is, the more complex is formed. And the reason more complex is formed is because more copper 1 plus, or more copper 2 plus was reduced to copper 1 plus. And the reason for that is because the more protein we have, the more copper gets reduced. So you can kind of see that little cascade right there. So really, the more purple that we see our solutions, the more protein is present. So here you can kind of expect that as we go up higher, the tubes get darker. And now we can compare our tube to this standard, and we're able to see that ours was about 6 mg per ml. Of course, we don't actually know that until we run the experiment. But that's just the general gist of how the BCA assay works. So normally I would say that's the end, but I want to go over a bit of housekeeping. So I don't know if you guys know this, but the course drop deadline is December 3rd. I have updated all of your grades. I'm just missing the lab report for labs 6 and 7. But other than that, everything should be updated as of now. After the BCA assay, we still have 2 labs left. I believe it's the activity and kinetics assay. And then finally, we will just have a final presentation. So I've decided that the final presentation will be due after Thanksgiving by November 30th. And I just want to talk a little bit about that. So what each of you are going to have to do is you're all going to have to record a 15-minute mock presentation and submit it to me by November 30th. So these videos should include everything that you've learned from the entire semester, starting, I guess, really at lab 2, so that's the PCR lab, to the kinetics lab. And the way that you're going to basically present this information is as if you were going to give a poster presentation to other fellow researchers. So this means that I really want to see that you understood the basic idea of what was done in the lab and really highlight the theories that you learned from the experiments. So this means don't just read off of your materials and methods. I really want to see that you understood what PCR is, how does it work, and why do we use it, things like that. So while you guys work on this presentation, I will try to have a rubric out for you guys so that you guys will know what info I will be looking for when I'm grading these final presentations. But other than that, if you guys have any questions, just please feel free to email me and I will try to get back to you as quickly as possible.